All right, we have hit noon. Good afternoon or good day, everybody, near and far in our digital auditorium. Uh, I welcome everyone and want to remind you that this video conferenced event will be recorded, archived, and excerpts may be used for educational purposes. By participating, you indicate your consent to recording, archiving, and use for educational purposes. So I want to give thanks to our sponsor at this last of our academic year's U of T monthly transfusion rounds. We've been supported by Alexion, CSL Behring, Grifols, Octapharma, Ortho Clinical Diagnostics, Immucor, Takeda, Instrumentation Laboratory, and last but not least, Orbcon. It is my great pleasure to be introducing Professor Dr. Jeannie Callum today. She's a transfusion medicine specialist and hematologist, an internationally recognized expert in best transfusion practices, safety, and multi-context hemostasis. She's been a beacon as the lead investigator on numerous high-impact, high-profile RCTs, fibers, FAIRs, and Concor, which is going to be discussed today. And she's the chief architect and leader of the Quest Research Group. She's also been an inspiration to the many people, myself included, who've entered the field of transfusion medicine and enhanced the knowledge of non-experts too with the wildly popular Bloody Easy books. Her brilliance began at Queen's in her undergraduate years and flourished in Toronto, where she attended medical school and postgraduate training in internal medicine, hematology, and transfusion medicine. Dr. Callum progressed to her place in the distinguished ranks of the University of Toronto with her leadership and practice at Sunnybrook. And last year, she returned to her alma mater, where she now holds the position of professor and director of transfusion medicine at Queen's and Kingston General, while keeping close to the Toronto family in our digitized times, forging and empowering wider research relationships in our provincial and federal community of practitioners. So with that, Jeannie, take it away.
Okay, thanks very much for that kind introduction. And I've been asked to speak today about convalescent plasma for COVID-19. And obviously this presentation was booked a very long time ago. It doesn't seem like that long ago, with the hope that we would be able to disclose all of the results from the Conquer-1 study, which I can't completely do today, but I can give you sort of the top line results that we've already announced. So we've come a long way in the last 14 months. Just for disclosure, my funding. So I'm going to go through first a history lesson on convalescent plasma. It's just really fascinating. Then I'm going to give an immunology 101. This is sort of what I learned during COVID. Then I'm going to talk about the US approach to giving convalescent plasma and where perhaps they have gone sideways. Then I'm going to talk about the UK NHSBT recovery trial and what they found in a very large trial. And then I'm going to discuss, well, why did number 3 and number 4 come up with completely conflicting results and why that might be true. And then that leads nicely into how Conquer-1 can answer question number 5 and the status of that trial. And then the lessons learned from the Conquer-1 trial. So I would want to say, like, what did I learn in the last 14 months? And what I learned is the immune system is super complicated. And that was really taught to me by Philippe Begin, who's one of our 3 co-principal investigators for the Conquer-1 trial. So let's start with this history lesson. So in 1907 was really the first publication. It came out of Italy, and it described the use of using serum from recovered people to give to people that were sick with measles. And there were 2 cohorts of patients, one in 1901 and one in 1906, but they were written up at the same time. And these were little kids with measles. Essentially, they took 600 ml of blood, whole blood from a 20-year-old donor who'd had known that he was recovered. They coagulated it, took the serum and put it in 3 tubes. I don't know what kind of tubes. They must have been very large tubes. They tested the serum first on a rabbit. The rabbit was okay. So then he tested it on himself. And then they gave it to 4 kids with household exposure. Nobody got measles. And then he repeated the experiment in 40 children in 1906 and nobody got measles. So clearly it works. And really that's how convalescent plasma is believed to have started. And then very quickly in 1915, there was an outbreak of mumps in New York. It was used again. And then for polio in 1916 in New York, and then for measles in Tunisia in 1918, people really got on board with this right away. And one of the largest early studies was from 1931 in a hospital in New York, in Brooklyn, where they had 180 kids in what sounds like, reading it now, is an orphanage for Jewish children. And they gave 11 children 12 cc's of whole blood. I'm assuming they probably gave serum. They don't talk about the blood group that had that from an adult that had mumps. And then as the kids got mumps, they took whole blood from the recovered kids and gave it to other kids to try and mitigate this exposure. And what they found was that the little kids that happened to be on certain wards where they were giving this convalescent plasma, I don't know how you were assigned to wards or whether that was random, kids that got it on certain wards had lower rates of going on to develop symptomatic mumps. And then, of course, in influenza, it was used, but obviously not in a controlled setting. This is one of the first ones published by McGuire in 1918, showing that, most people did really well if they got serum from people that had recovered, but not in controlled studies. In 2006, Luke et al. Went back and looked at the studies from 1918, and they suggested that in these uncontrolled studies that the rate of death from convalescent plasma was lower at 16% compared to 37% if you'd been lucky enough to get convalescent plasma. And it was better if you got it early than compared to if you got it later. Obviously, that's a very biased analysis, because lots of people that say got it early would have gotten better on their own. In that Luke report in 2006, this is what the forest plot looks like. And all of the estimates are really looking like it favors treatment, and everything to the right of the zero line, suggesting it's beneficial. And then right when COVID was launching off, Philip Begen's group released this on May 22nd in CMAJ, looking at not just the influenza trials, but also SARS and Ebola. And it didn't look quite as impressive as those early non-controlled studies, and that's commonly seen. So it didn't look like there was any mortality benefit as shown in this forest plot with everything crossing that one line. But days in hospital and days on the ventilator looked a little bit better in these studies. And so there was a lot of hype and excitement about using convalescent plasma based on this and other systematic reviews. Okay, so that's where we started before all these trials started going on. So before we talk about these trials, you need to know a little bit about the immune system and the testing that went on the donor antibody profile that was being put into these COVID-19 patients. So you can measure the antibody levels in the donors in multiple different ways, but there are 4 common tests, and all 4 of these were done in the CONQUER-1 study, so that we can correlate outcomes for donors, for the patients that got blood from donors that had high levels of one, lower of another one, and does it matter what the profile of the donor was? So you can measure antibodies against the spike protein, the whole spike, there on the left. And in a titration kind of model. You can also measure antibodies to this receptor binding domain, which is the part of subunit 1. And that's thought to be more neutralizing, and it correlates well with your neutralizing antibodies. So you can measure that again in a titer. You can also measure the antibody-dependent cellular cytotoxicity. So this is correlating IgG antibodies, how well they bind to your target, and then your natural killer cell able to bind to your IgG, cross-link those receptors to activate your natural killing cell to make it kill the target. So you can measure this in assays of your donors. You could also obviously do it on your recipients. And lastly, this what's called a PRNT, or a plaque reduction neutralization test. This is also like the ADCC, it's a functional assay. So in this assay, you plate your Vero cell, green monkey cells, into a plate. You add your antibody in a dilution, and then you infect the cells, and you see how well your antibodies protect the cells from the invasion of the SARS-CoV-2 virus into those cells. And the plaques are sort of the dead cells. And so you can see these white spots. And then you correlate, when does it protect greater than X amount? But usually it's a 50%. And so some of the studies used a cutoff of a titer of 1 in 80 or higher. Some used a cutoff of 1 in 60 or higher. That's what Canadian Blood Services did in our Conquer-1 trial. So they chose this much higher titer. Some studies, like the recovery study, used a cutoff on their RBD antibody that correlated in their lab with a cutoff of around 1 in 100. And so people have tried to, use this as kind of the gold standard of the testing for your donor. But even before we started, this is an article from 2003 that suggested that maybe more antibody might not be better, and that always giving more of something in medicine isn't always good. So obviously you can give too much of a chemotherapy. Similarly, you can give too much of a good thing. And in their model, that they had a mouse model of Cryptococcus, and they adjusted the types of antibodies they were infusing into these mice, as well as the infection inocula, to see which combination resulted in higher lethality or better protection. And they found that some antibodies increased the risk of dissemination into the brain, so it actually made things worse, and that individual antibody combinations could be protective, non-protective, or disease enhancing. And so this entity was called into almost like a prozone effect or a hook effect that we might see with a laboratory test, and that there could be too much of a good thing. That hook effect is related to this. It's called a hook because of the shape of this curve. And this hook effect relates really to laboratory tests. So for example, this is a test result on a woman who has a very high level of HCG. But because the excessive amount of HCG overwhelming the binding capacity in this ELISA assay, looks like it gets hooked at the result at this around 6,000, but really if you diluted it out and then remeasured it, this woman's level is actually over 1 million. And that's called a hook effect or a prozone effect. And so there's the thought that perhaps this happens in medicine as well with convalescent plasma. In addition, we have results from HIV vaccination trials. This is a randomized trial from 2009, randomizing people not HIV infected to a vaccine and comparing the rate of developing HIV. And you can see there's a trend towards a reduction in HIV prevalence, but you can see right there on your y-axis, this is just on a percent. So it's just a fraction of a difference, so not terribly effective. But what was interesting, and that was published in the New England Journal, was that if your vaccine resulted in you making a very high IgA antibody compared to your IgG level, you would actually increase your chance of acquiring HIV infection. And so this is your odds ratio around 1.5 if you made a higher IgA compared to your IgG. And here on these graphs just shows on the left, the black curve for acquiring HIV is in black. In purple there is your reduction if you happen to make a good IgG antibody. But on the right, you can see you actually do worse if you make a high titer IgA compared to, it would be better not to have had the vaccine. So our response to the vaccine or our response to plasma might be different compared to what kind of antibody profile we have. So obviously the immune response is complicated. Okay. So with that in mind, let's talk about what the US did for their approach to convalescent plasma. So on August 23rd, the FDA issued an emergency use authorization, a little bit pressured by the president, for COVID-19 convalescent plasma for the treatment of hospitalized patients with COVID-19, and citing that there was such clear evidence from historical reports from the Spanish influenza, from other infections, that we didn't need clinical trials. We should just go ahead and start using it. And not everybody liked that messaging. So here's a headline from Stat magazine. We don't know if convalescent plasma is effective against COVID-19. With the emergency authorization, we might never know because we're not going to have a control group. So not everybody was happy with this. And then in February, so that's quite some time later, in 2021, they put in new rules, and they required that only high titer plasma would be used. And so they set a cutoff based on these tests, like the orthovitreos. So remember that, greater than 9.5 and the Euroimmune greater than 3.5. These aren't super high cutoffs. These aren't neutralizing titers above 100 or 160. It's just not very low. You had to use it early in the course of the disease, before intubation, or in people with no humoral immune response to the virus, somebody that maybe is immunocompromised, and you have to label the units as high titer. So, you might think, I wonder what triggered that. And it was really this article here. So this is an article about convalescent plasma, antibody levels, and the risk of death from COVID-19 from the US program. So remember, there's no control group in the US program. And this was really led by a combo team of Michael Joyner and Arturo Casadevall that led this. So they reported that in the New England Journal of Medicine on adult patients with at least one risk factor for severe COVID-19. So those are risk factors they were doing badly from a respiratory point of view, and those criteria are there on the right. They gave 1 to 2 units of convalescent plasma from 54 blood collection centers, but they only looked at patients that got a single unit for their antibody analysis because they didn't know how to combine if they got in 2. So I'm not sure how valid that is. There was no control group for this study. There was no threshold to qualify the donors. They divided donors into 3 categories: the bottom 20% on the orthovitreos test, the top 20%, and that middle 60%. So cutting them off at the 20th and the 80th percentile. They had 3,000 patients getting blood at 680 sites across the United States. But so far, over 55,000 Americans have been treated with convalescent plasma as part of this EAP, or then on label as part of approval by the FDA. They didn't get quite a perfect balance. So if you looked at the high titer patients, not quite so many were intubated, just a trend. The number of patients that had complete whiteouts, more than 50% prior to treatment, or a low oxygen level, were just a little bit lower in the high titer. So the high titer group possibly looked a little bit better than the low titer group. And what they found was about 27% of the patients died, which is, a hospitalized population that was treated with convalescent plasma. But the low group, about 30% of people getting a low antibody level died, compared to about 22%. So about an 8% absolute difference in mortality if you happen to be fortunate to get that high titer plasma. So the relative risk reduction comparing high versus low was a 0.75 and statistically significant. So these are large numbers of patients. That's probably a true estimate that high titer might be better than low titer. And here's in their model looking at low titer, medium titer, and high titer, and they set automatically the reference as the low titer, assuming that more must be better, which is unusual because Casadevall, who is the senior author on this paper, also wrote that article about the mice with Cryptococcus, saying that more might not be better. So you can see here they use the reference group as low titer, but you easily could have made the reference group the high titer with the assumption that, is it possible that low titer might be harmful and high titer doesn't do anything? This was a random assignment, or you could have made the middle one the reference population. Okay, so with that in mind, probably a true fact that high titer is associated with a lower risk of death compared to getting a low titer, but no control group. Okay, let's look at a study that has a control group, the UK recovery trial. So this is a study coming out of the UK of 12,000 patients randomized to convalescent plasma compared to standard of care. The CCP units used from the NHS had a high titer, so about a neutralizing titer of 1 in 100. They qualify based on this Euroimmune, which is like the orthovitreous ELISA test. And so high titer compared to nothing. Most of these people were not vented. Most of them got corticosteroids, which we know reduces the death rate. Median time from onset of symptoms to treatment was 9 days, pretty short. not too long, and they got 2 units of plasma over 2 days, and their primary outcome was mortality. And there was absolutely no difference. So on the left there, you can see the survival curve, and the odds ratio is 1.00. And it didn't matter which group that they looked at when they looked at their subgroups by age and gender, sex and ethnicity. There appeared to be no difference. A lot of people put a lot of hype into these two estimates for people that got treated early versus people that got treated later, suggesting that again here, if we treated people earlier, maybe it would actually work because the estimate was 0.92. But obviously hypothesis generating, not proven, but a lot of hype went into this particular graph from the paper. For those transfusion people, particularly for Christine Surti, I thought you would be interested in this. So in the recovery trial, if you go into the fine print, you'll see that they had 13 transfusion reactions out of 5,700 treated patients, 9 respiratory and 4 other, for a reaction rate of 0.22, with 3 deaths possibly related to transfusion. All were pulmonary reactions. And so I wonder, we know that the rate of TACO is about 2%, 3% for patients getting plasma. Is it possible that in these higher risk, mostly older patients, that you could get down to a TACO risk of 1 in 644? Or are we suspicious that perhaps poor transfusion reaction reporting happened within the recovery trial? So in the recovery trial, they actually added a figure 4, and they updated the meta-analysis of all the other trials. And you can see there that the risk of death, whether you got convalescent plasma or not, including all the previous smaller randomized trials that went ahead, there's no difference. It just doesn't work across the board. This is in comparison to another meta-analysis that was published a few weeks before that one. And you can see there that they find an odds ratio of 0.65. So it's all about how you spin the data. So lots of people are still trying to say that convalescent plasma is favorable. Since the recovery trial happened, 2 other, I guess a 4th other, if you include the CONQUER-1 study, was terminated early for futility. So the first one that closed for futility was the REMAP-CAP study. So this was ventilated patients a little bit farther into their treatment, no benefit. This is not yet published yet, but hopefully will be published shortly. So it didn't work, and it was a large trial. In addition, another trial, and I've got C3PO, I keep looking for R2D2 trial, but it's a C3PO trial. This was an NIH-funded trial that was early treatment. So these were patients coming to the eMERGE with high-risk features, given high titer, antibody neutralizing level above 1 in 160. So very good plasma, very early, very large trial, no benefit. So I guess my question for you is, how is it possible that we have recovery, REMAP-CAP, C3PO, and CONQUER-1, 4 large randomized trials that find no benefit, but then we have Joyner et al. That finds a significant mortality benefit with high titer convalescent plasma? So how is that possible? So, with the stoppage of the recovery trial and the REMAP-CAP outcomes in Bloomberg, you know, the Trump-touted plasma therapy shows no benefit, researchers say. But at the same time, we have other messaging going on. This is again by Dr. Casadevall, and this is a preprint that's out there from April 10th. Did plasma hesitancy cost lives in the United States? If we'd used it more aggressively, could we have saved more lives? So completely different messaging. Including very large and big stories about how scientists had revived old school treatments for the 21st century pandemic and how effective it was. And here's, you know, Johns Hopkins University. So I'd like to take you back to about grade 4 of mathematics. And you might have had a test like this, something like if A is equivalent to B, and if A is greater than C, then it would be obvious then that B must also be greater than C. And that's really just a straightforward mathematical proof that most of us can easily see. And I think that this is very similar to what we're seeing with convalescent plasma. So we have a pretty good truth that there's no mortality benefit in 3 large trials using high titer plasma. These 3 trials did not use low titer plasma. So they can't compare what low titer plasma might do. And in contrast, we have a really large study that shows that statistically significant, if you got high titer plasma, your mortality rate was lower than compared to if you got low titer. So thus we know that high titer is the same as nothing. We know that low titer is worse than high titer. Thus, by mathematical definition, low titer must be worse than nothing. So the conclusion must be that low titer plasma is harmful to patients. And so this is what we need to figure out, whether or not this is true. And how could low titer or bad antibodies cause a problem? So, patients that come to hospital already have some of their own neutralizing antibodies. And if you infuse plasma with a lot of non-functional or low levels of antibodies, you might make things worse. Your non-functional antibodies might block the binding of functional antibodies, making them worse. Some donor that say makes IgG subclass IgG 2 or 4, that's not going to give you very good ADCC, or they make high levels of IgA or IgM, are going to interfere with your ADCC function. And so obviously this is pretty complicated, taking plasma from any donor and put it into any recipient without understanding what either the donor or the recipient immune response is doing is pretty far stretched. But one of the great things is, fortuitously, the Conquer-1 study can verify what must be true from these studies. So we had a 2-to-1 randomization, so twice as many patients got convalescent plasma compared to standard of care. And we have 4 blood suppliers, the New York Blood Center, Hema-Quebec, CBS, and Hemo-Rio. Only one of them used high titer only. That was Canadian Blood Services with a titer of 1 in 160. And the rest of them used a mix of low, medium, and high. And so we can look at, well, how did that difference, because the other 3 trials only used high titer, and we had a nice mix. In addition, under the leadership of Philip Began and his big immunology team, we were able to do 2 functional assays and 2 quantitative assays to understand how does that impact the mix. And then yesterday, telling us that the antibody study is not over, announced by Recovery, that the Regeneron monoclonal antibody combination, so remember, Regeneron is what Trump got. This is 2 monoclonal anti-spike antibodies. They're mixed to try and get rid of some resistance that might happen. They randomized almost 10,000 patients to antibody combo versus usual care. And in the third of the patients that were seronegative at baseline, yesterday they announced that the mortality rate was half if you've gotten the Regeneron. So there is some suggestion that we're not done with antibody treatments for patients with already active infection. Okay, with that, I'm going to transition to the Conquer-1 for the last little bit. So what do we do in Conquer-1? So this was a multi-center, multi-country study looking at whether convalescent plasma would reduce the combination risk of intubation and death at 30 days for those patients admitted to hospital with COVID-19 compared to standard of care, excluding patients that were already intubated. You can see here our eligibility criteria were essentially adults admitted to hospital on oxygen that we had convalescent plasma available for them. Alan Tinmouth and Mickey Zeller, CBS, and the MCTR group did such a great job that very few patients got excluded because we didn't have ABO compatible convalescent plasma. And then you can see there minimal exclusion except for the first one, and that was beyond 12 days from onset. This was the investigational product. So this was plasma collected by apheresis from someone that had recovered from COVID-19. Each patient received 500 mils, preferably from 2 separate donors, and given a standard way. They were randomized, as I said before, in a 2-to-1 allocation, and each plasma was infused over 4 hours in our hope that this would reduce infusional side effects like TACO. Primary outcome was intubation or death at 30 days. We chose this because we were worried that some patients would not get the opportunity to be intubated because we would be overwhelmed by cases and would not have enough ventilators. We collected also secondary outcomes to look at efficacy and safety that will be reported in the paper. Remarkably, there were 72 hospitals across Canada, the United States, and Brazil. It really was an incredible feat that in a very short period of time, this trial was planned, launched, executed, and data entered, and the paper submitted for publication in under 13 months. So our current status, we had a planned unblinded analysis by our Data Safety Monitoring Board at the halfway mark. At that point, there were 614 patients with complete data, and it met our pre-specified futility criteria. The conditional power estimate was 1.6%. We were going to stop if it was below 20%. So there was no way that we were ever going to show that convalescent plasma was beneficial. By the time the trial was stopped, because of course we kept enrolling patients, we were at 940 of the planned 1,200 patients that would be enrolled. And the data entry now is complete. The sites did such an amazing job getting the data in really quickly. And obviously we can't talk about the results until it's published. But we saw futility. We will be able to look at those antibody levels as well as subgroups of patients depending on the donor profile for those 4 antibody tests. We had incredible support from dozens of funding agencies, both peer-reviewed and non-peer-reviewed, and in-kind support from multiple people to make this trial happen. And really Conquer One was this huge achievement for the transfusion medicine community across Canada. Probably most of the people on the line today participated in some way for this trial, thawing the plasma, issuing the plasma, randomizing patients, screening for patients, talking to patients. Really well done. It just shows that across Canada, we can do multi-center trials and answer some pretty important questions. Okay, so just one slide on things that we learned. Because people were obviously worried about we were all going to die of COVID, there was incredible dedication by everybody across the country that we were going to see if we could figure out if the transfusion medicine community could contribute to understanding how we help patients. We had 3 co-PIs. I think if we'd had one of us would have melted. It took 3 of us not just to do the workload, but also the moral support, ethical support, moral, emotional support during this trial, which was really difficult. We had a very large steering committee with microbiologists, infectious disease experts, statisticians to really help us. We had a really experienced, international experienced data safety committee to make sure that we were steering the trial in the right direction. We divided the task across multiple people across the country to get the trial launched. We had numerous statisticians under the leadership of Richard Cook, who's honestly brilliant, from Waterloo. We included non-academic hospitals that were harder hit. Of course, we weren't going to know this at the beginning, but they were harder hit with COVID. And if we hadn't had them, we would never have been able to complete the trial. We had a community engagement committee that took over like the messaging and how to talk to the public about the trial that you don't normally have to do while you're doing the trial. It's just after you finish it. We had the support of the 4 blood suppliers even before we'd achieved funding. We had experienced virology and immunology labs that were recalibrating every time a new test or a new assay could be done to add it on to the donor profile so we could have it for publication of the results. And Health Canada, the FDA, and the REBs honestly approved this study almost overnight. And all the digital platforms really allowed us to run this trial without anyone leaving their offices, including most of the consents done for the trial were done by the phone or through some device. But we did have lots of challenges. The first was, we knew that convalescent plasma was going to have to be tested in a pandemic, but we never came up with a protocol or how we would manage the inventory. And maybe we planned for blood shortages to do with the pandemic, but we should have planned for this because we knew it was coming. It was talked about when SARS happened. There were trials during SARS. So we should have planned for this. We should have planned for an integration of the pandemic clinical research staff on the patient facing, because a lot of these studies, the RAs were lining up outside the patient's room and do you want to be in this trial? Do you want to be in this trial? And the patients were kind of overwhelmed. They didn't know which trial should I choose. We should have engaged better with the clinical and the laboratory staff to develop strategies to enable research during a pandemic. We should have opened 100% of the sites, even if they didn't have cases before the pandemic hit. A lot of people sort of said, oh, we're not going to open, COVID's not coming to us. We haven't had any cases. And then their center blew up. And so we had to open them in the middle of, that additional chaos. We had centralized REBs in Ontario and Quebec that allowed us to launch those two provinces right away, but some of the other provinces didn't have that. And I think that that's really, we need that. And we also need centralized contracts rather than trying to do data sharing contracts hospital to hospital. It just takes too much time during a pandemic. And obviously we need a lot more research into our immunologic response to viral illnesses. If we're going to use antibody treatments, we need to understand what is the variability person to person. So to summarize, convalescent plasma's had this very long and interesting history. And this is the first time we've been able to answer that probably it's not very useful right across the board. As I've learned during COVID, your immunologic response is super complicated, and it's too simplistic to say you can take half a liter of plasma from one human and put it into the other. We talked about the US approach to using convalescent plasma without a control group, and maybe that wasn't the right decision. And we probably could have answered this question 50 times over with so many patients treated with convalescent plasma in the United States. The UK recovery trial has really shown us, along with C3PO and REMAP CAP and Conquer-1, that it actually isn't better than standard of care. We talked about why 3 and 4 might have conflicting results, and that's probably because low titer plasma is harmful rather than high titer plasma being better. And we talked a little bit about the Conquer-1 trial, and I'm hopeful that very shortly you'll be able to see the results of that trial and all the hard work you put into it. And then we talked about the lessons learned for the next go around. If we ever have another 10th wave or another pandemic, I think we're going to be a lot farther ahead in our ability to do research. And with that, I'll stop, and I thank you for your attention. I asked Donnie Arnold and Philippe again if they could attend if they weren't stuck in their clinics as well, to answer any questions about the trial. Thank you.
Oh my goodness, Jeannie, that was enthralling, masterful, virtuosic. Wow. What a thrilling presentation, and what an interesting debate in space-time, given the Evan Block talk 6 months ago in December 2020. That was incredibly enriching, and we thank you for the incredible work of the trial, as well as the information distillation, including down to the level of 4th grade understanding. Absolutely awesome. I do want to remind everyone to take a moment to complete the feedback survey in the link in the chat box and at the completion of the presentation. Obviously, your valued responses help us in future rounds. We're going to open up the floor for questions. Comment from Dr. Katerina Pavensky, great presentation. Also from Dr. Yulia Lin, amazing presentation and overview of the trials. Jacob Pendergrast, Dr. Jacob Pendergrast is waving.
Hey, I can never find my little hand icon, so I'm just going my actual hand. Jeannie, that was so interesting. Your one reference to the concern about plasma hesitancy sort of raises the idea of contrasting the experience we had with convalescent plasma with vaccine development, which obviously was more successful in hindsight. And I'm sort of wondering, is that a reflection of the fact that that we didn't have enough time or enough resources to do all the preclinical work that we need for convalescent plasma before we launched right into an RCT? Like, if we could go back in time and do some of those, more elaborate, laboratory or animal studies to kind of figure out if we knew exactly what was in this stuff and what the therapeutic action was, would that have helped, you think, or was it just something that we just can't do with this kind of therapeutic?
Well, I think actually where we went wrong was, we see influenza every year. It's really predictable. Unless we completely 100% mask, like the last year, we get lots of influenza. And there is no reason why we couldn't have done these trials in influenza, both in animal models and then in human models, and gone through this whole trajectory. So at least we would have been a lot farther ahead in our understanding of our response to regular influenza. There was no reason why you couldn't have, as healthcare workers get sick, you're swabbing everybody. Find your influenza donors and your healthcare practitioners every time they get a cold. And we could have done this. I'm not sure, looking back, Rob Fowler during SARS, he was like, Jeannie, we need to do a convalescent plasma trial. Like, I remember this, but SARS went away so fast, and then we're on to something else. So always, look ahead. Like, I think it's really important to look ahead into time and, help your older self when you're, planning these things. There's a question there from DT about the Argentinian trial. So it's really interesting. So the Argentinians did a trial that they stopped early because they had, not enough recruitment, and they gave it early to people out of hospital, and they saw a big difference. I think it must just be some sort of random chance, because the C3PO is way bigger, very high titer. So I think that whatever they found can't possibly be true if the C3PO just shows such a nil effect.
So more praise from Rebecca Barty, Joanne Duncan. A question from Katerina Pavensky. Difficulties encountered trying to publish results. Comments.
Yeah, where Donnie Arnold was there for a second, and then his camera's turned off. I don't know if he's. Oh,
I've
you're
got
right
Donnie
there.
Arnold's hand, but muted.
Yeah.
Yeah. There we go.
Donnie, do you want to answer the?
Sure.
Trying to publish in the land of Republicans and Donald Trump trying to say that convalescent plasma isn't helpful.
You know what? One thing I've learned about doing research and publishing papers during COVID is that it is so much about timing and what journals want to take up and how newsworthy it's going to be. And that's really a shame, because that's really not the reflection of evidence, right? I mean, Twitter has become kind of the mainstream source of information for lots of folks, and that I think that's relatively dangerous. And for journals to just take up things because they're super hot at that moment does not do the scientific world justice. Now, having said that, so we're facing that that tidal wave right now, where plasma is yesterday's news. I mean, that's just really what people are thinking from the journal's point of view. But this will get published in a decent journal. And the fact is, the impact factor, or whatever metric you want to use to measure journal value, is probably irrelevant or less relevant than getting this message out. And this trial has so many messages. It's got a randomized trial message. It's got a translational focus on antibodies. There's so many other messages that are going to come out of this. I think this is going to be such an important contribution to the literature and to evidence, no matter which journal it finally ends up in. But I'm still optimistic it will end up somewhere good. But it's been a really interesting kind of venture to see this happen. I had a question for Dr. Callum, however, though, if I can ask one. So given what we now think we know about the potential, I think your algebra is probably true, but there's potentially danger to some of the plasma units, like they may be harmful to some degree. The entire world is going to be vaccinated or have seen COVID or whatever. So every unit of plasma that we now administer is going to have some anti-COVID antibody in it. And, lots of people have had influenza, lots of people have had X, Y, and Z infection, but we're just using plasma for lots of different reasons. Is it possible, do you think, if you want to kind of just guess about this or pontificate about it, do you think there are some dangers to plasma that have gone unnoticed up until now?
Yeah, so the first thing is even before our say if you did a trial with plasma before the pandemic, there are 4 coronaviruses that circulate every winter. So if you're an adult and you've had kids in daycare, you're going to have pretty good coronavirus antibodies, but they're not really neutralizing, they're just distracting. So even our regular plasma, our IVIG, has that base amount of IgG that might be, or IgA, or whatever the antibody is, might be distracting. And now we've got basically everybody's going to have some difference of Moderna antibody response versus AstraZeneca response, going into a mix of your IVIG and your plasma, so that for the near couple of years, basically every blood product we're transfusing, whether it's platelets, plasma, is going to have, all these antibodies. And we have absolutely no idea what that's going to do to your infection risk. Is it going to make it lower or higher? What's going to do to your antibody response? We don't know. It's kind of a frightening, we're in like basically a black box, and we have no idea what's coming. Particularly, I think, for the IVIG. We had our first patient refuse blood products like a Jehovah's Witness because she was afraid that the blood products would have antibodies from somebody that was vaccinated.
Mm-hmm. Yep.
So I think there's lots coming to be studied on this, and that's and one of the good things that's going to come out of the Conquer 1 is just the recipient antibody testing. What was the status of the recipient at the time they got the plasma, and what was that? How did that impact on their response to the treatment? And those samples are still frozen. They have not been tested yet. So we have a whole viral load on those donors and their antibody levels to see how that, mixes into this question.
So, it's very interesting, the notion of titer. Another notion of how much antibody you distribute in terms of amount is also the intensity of that distribution over time. So timing has been it's felt like this kind of really random undercurrent. It intrigued me that in the recovery trial, they gave those 2 units over 2 days. And then when you're getting, these passive antibody concentrates, it's infused over a shorter period. We gave our 2 units, within a tight period, within a single day of, the day of enrollment. Do you guys think that there's anything there in the time angle of different outcomes and study designs according to timing?
Yeah, I think if you look at all these studies, they're ever so slightly different. One of the things that I don't think we really understand is you're giving 2 donor bags, one from donor A and one from donor B, and they might have completely conflicting risk and benefit, and you're kind of mixing them together. So was it better to put them together really closely so if one could negate the effect of the other, versus if you spread them out, they're almost like an independent effect that was done in recovery. In addition, some of the trials actually used controlled plasma versus just standard of care. And some of them, the one that did do it with a controlled plasma, showed a benefit, but it's possible that's because the control plasma was worse than nothing, because you would need to do a study with nothing, regular plasma, and then convalescent plasma to actually answer, does this regular plasma make people worse? Remembering that all plasma's got some coronavirus antibodies from our head colds.
Yeah, when you've got something this complicated and that many theoretical arms for true comparison, you need animal models, you need something else. It's just really hard to execute in humans. Katerina Pivensky has her hand up.
Hey, I just want to pick up on something that Donnie said, and I have to say that this was, we recently had our CSTM annual meeting, and there were entire sessions of that meeting completely dedicated to COVID-19 and immunology, et cetera. And I was moderating one of such sessions, and I remember walking out of there thinking, okay, I'm not sure if I understand 100% of what everything was being said, but what I'm walking away from is that notion that Donnie just alluded to, is that plasma may in fact be harmful, and in fact, the majority of donors in Concord trial were not the quote unquote good donors. They were the ones who were potentially giving you the antibodies that may, for the lack of better word, distract your immune system from either mounting an appropriate immune response or effecting an effective immune response. And then there was this big discussion about if you did a trial, and I'm not suggesting, God forbid, that we do a trial, please, but I wonder, in view of that, if you, instead of using people who had natural infection to COVID-19, you used as donors people who received their 2 doses of vaccine. We know vaccine is 97% effective, which means that the people who received the vaccine would have had the highest titers of the bestest antibodies available. Would your result have been different? Or is this just, we're completely, should be shifting gears and forgetting about plasma and thinking about other therapies? Anyways, I just wonder if either Donnie or Jeannie, you want to comment on that.
I think one of the things that we learned is that plasma from a human is a biologic with a distribution of your ADCC, of your anti-RBD, your neutralizing antibody, and it's a complete mix. And every single donor is its own unique milieu. And if one antibody is protective, another one is harmful, you can't separate the two in a plasma product, because it's always going to be a mix of these different antibodies. So it really, in my mind, helps inform that really in a pandemic, you've got to go with a monoclonal designer antibody. It's got to be IgG. In your animal models, it's got to have high neutralizing, high ADCC before you go on to test that in humans. That the Regeneron, that kind of model. In addition, somehow you got to figure out how to test your recipients. If the recipient already has a great neutralizing antibody, don't mess with that patient. Restrict your trials to people that are not making neutralizing antibodies, not making any antibodies. And so I think for the next pandemic, for some future generation, they at least kind of have an idea of where they're going to go next with this. If you're immunocompromised and you get COVID, your outcomes are terrible. And so I think it's going to be really important that we do trials in that patient population.
No, I'm really fast. Oh, sorry, go ahead.
Sorry, no, I was just going to add to that. I think you're right, Katerina, that if you took vaccinated people and collected their plasma, you're probably more likely to find people that had high titer antibodies. And I think, but I'm not sure, would meet the profile of functional antibodies as well, being on the higher scale. But I totally agree with Jeannie. I think what we're learning is that plasma is like a soup. It's got lots of stuff in it. It's not very specific. And there's all things that we're not even considering that are in there too, like whatever other proteins and cofactors and cytokines, et cetera, that none of those are being controlled for either. So it's really tricky to have a standardized product when you're doing a clinical trial in a human biologic product like that.
To link all these thoughts, it makes you wonder if these low titer bags or these control plasma arms or what have you are really incursions of those unmeasured reactions. There's no way that the reaction rate is less than everyone else, and that this whole having COVID and getting plasma actually puts you in a position of a lower than average risk of transfusion reactions. That's absurd, right? And so the imputability problem becomes this blending fiasco of, how much of this is the underlying condition and how much of this is additive harm from that particular donor's soup, the anti-interferons, the subverting or interfering antibodies, maybe even an Rh immune globulin Amos-like effect, the antibody-mediated immunosuppression, where you steal that recipient seroconversion thunder. So how much of this is slow scale, additive transfusion reaction versus later scale harm? It's raising questions also of like follow-up effects, like who even knows what the effects are many months out in recipients, and the different variants of concern and latter waves. So I think that there's so much to unpack in Concord, and it's such a treasure trove with what's been bioarchived. I think what we're going to learn from the way that you've designed this and all of these collateral studies is going to be incredible. So it's very exciting.
There's a great question there for Canadian Blood Services. I don't know if someone from CBS is on, but will CBS be collecting information about who's had infections, who's been vaccinated, so that could be logged, and we could look at doing, kind of like phase 4 studies, looking at recipients of COVID vaccinated recipients versus not.
I see a response from Dr. Aditi Khandelwal.
Hi. I can partially answer this question because we know that this is definitely a topic of active interest and research at CBS, and some of my colleagues are working on this. But I can tell you that in the donor screening process, everybody's asked about history of if they've had COVID in the last 14 days, because that is a cause for deferral. And we do ask people not to come in if they have any symptoms. And even before entering the donor center, people will go through the screening like many other places that people go to. For the vaccination piece, there is a question in the questionnaire about vaccination. This question actually was originally put in there. It's not a new question per se. It's an old question because a lot of our patients are immune compromised, and live vaccination can be a problem in individuals receiving blood with potential contamination with live virus, right? This question also captures COVID-19 vaccination. Currently, we do not have clear evidence that having been vaccinated with any of the COVID-19 vaccines that are being used in Canada, actively used as well as under research, has led to any harm. But again, this is very new for everybody, and different countries have different criteria for multiple different reasons. And this is being studied. I do not know the way the study is structured, as I'm not part of the actual process of study. I will tell you, though, that some countries have decided that maybe a 48-hour or 72-hour period after vaccination would be a good time for deferral, because a lot of people do have the flu-like symptomatology after vaccination. And whether we should have people who've had COVID vaccine donate other products other than whole blood, so we're talking about stem cells or not, is also an active area of consideration and being looked at very carefully, because the considerations are slightly different in whole blood versus other product collections. So this is definitely a very hot topic. I wish I could tell you more, but that's the extent of what I know.
But great idea to do a look at, patient outcomes versus donor antibody status or vaccination status would be a great contribution, especially now that we're able to digitize the donor information and the recipient information.
Yeah, for sure.
All right, I can't resist near the end here sharing Melanie Tokeshi's comment. Dr. Arnold is right. It was a topical and sexy trial to be a part of. Everyone was more on board and more keen than any other ask that has come through the blood bank. So I guess trials matter. The success of this had a lot as well to do with, how people felt about this. We were all in a really scary global pandemic situation together. A question here from Fatima Hossein: is there any role of convalescent plasma transfusion without knowing the neutralizing titer of the donor in the low resource setting?
Yeah, so India did a trial, it's called the PLASA trial, where they actually did just that. They took plasma out of recovered people and gave it to people without knowing the antibody status. And a good, quarter of those units had no antibodies at all. So you definitely can't do that. And people had hoped that in low resource settings, that this might be something that they could afford. You definitely don't want to go there. You're probably going to hurt more people than you're going to help. So that didn't turn, that was a hope and a dream that this could be done in Africa and India and all these different countries, but definitely not.
But that's another reason why we feel such a moral imperative to get this trial published as quickly as possible, despite some of these challenges, because there are people in the world who are still using plasma because it's available. And it's not really good enough to say, well, it's no better or it's no worse than standard of care. It's not only potentially no worse than standard of care, it might actually be harmful. And I mean, we're not saying that with certainty. That's like a secondary exploratory analysis, but it's pretty compelling that there's a signal for harm.
On that note, I want to thank Dr. Jeannie Kellum, Donald Arnold, everyone who was a part of Concord, and all of our audience today for participating. From the bottom of our hearts and the top of our minds, we are so grateful for the culmination of this work and where this takes us further. Thank you so much, and congratulations and thanks to Jeannie for a really outstanding session. I couldn't have had a better talk to finish our U of T monthly transfusion rounds year. Thank you.